Okay, I think I might just go ahead and get started. This will be recorded. I'm gonna send the lectures out, or sorry, the lecture slides out by email later today. So I guess if anybody misses, they'll be able to catch up. Also, we can chat on Discord, but yeah, I was hoping we'd have enough time to get through everything. Oh, hello? Okay, sounds like everything's good. Yeah, please feel free to unmute yourself if you have a question. There's sort of a lot of you, so I might not see if your hand is raised, but I'll try to also watch the chat. But yeah, this is just incredibly exciting to have you guys all here. This is something that I think Kato has been dreaming about for a long time, and he's in Paris right now, so I don't know if he'll join the Zoom call, but yeah, this is like a dream that I've had for at least 6 months. So it's just, yeah, super nice to be here with you all and excited to chat more about synthetic biology. So I'll just go ahead and start by sharing my screen. So I've got the slides open. Let's see. Okay, it doesn't like it. Let's try again. Okay, what's going on? Let's do this instead. Very interesting. Okay, I don't know why I'm having so much trouble, but let's do this. To here, get out of this. If I share screen first, okay. Okay, you guys, I think now should see some of my slides. Let's see what happens, oops, when I go into slideshow mode. Okay, do you see sort of a big white square?
Yeah, sure, we can see it.
Okay, fantastic. And if I hit forward, you see the next slide? Now there's like some bolded words.
Yeah, sure.
Okay, wonderful. Well, thank you guys so much for being here. So I guess you are all here because you're interested in this topic called synthetic biology. And I thought it would be helpful to, I guess, just talk about what does that mean exactly. I mean, maybe many of you know, we're going to have a discussion later about kind of what the background is of people in the class. But yeah, hopefully, even if this is really targeted for people who've maybe never heard of this before or like heard of it a little bit and think it's exciting, so that we can sort of increase your excitement and then give you the tools to learn more, maybe pursue a career, something like that. So I thought I would start with just looking at this phrase, what is synthetic biology? And it has two key parts. So I think the synthetic part, what that means to me is basically it's when you've engineered biology in some way or you've changed it. And something that I think is particularly exciting is basically what you are doing is creating a type of biology that has never existed before. There's like really cool stuff where people have, for example, changed the way that I guess RNA molecules encode proteins. It's okay if that doesn't make sense to you yet, but basically we can change the very way that cells work. And then in terms of biology, a lot of what I think about and what I think is common is cells. These are like the fundamental building block of biological organisms. This can include sort of organisms made of individual cells like bacteria. There also are yeast, which are, again, like bacteria, they're sort of small and single-celled, but we tend to interact with them in the bread making process, or if you're brewing beer, yeast are what is responsible for turning the sugars into the ethanol, which makes the alcohol. But also something else that people are really excited about, particularly if you want to do something in medicine, is cell lines that are actually human cells. But you don't just have to be kind of engineering cells. You can also be engineering kind of the basic molecules of life. So this includes things like DNA, which you've probably heard a lot about, or like the human genome. There's also RNA molecules, which I think people are even more excited about now because many of the new COVID vaccines that were made were based on RNA molecules. And also there are proteins. So maybe you've heard of enzymes or I guess other kinds of proteins that can do important functions. And then something else that maybe people forget are also viruses, which are something else that we can engineer and work with. Okay, so that's sort of, I think, what synthetic biology means. Basically, if you're engineering, changing, or like building something, and you're using cells, DNA, RNA, and proteins. Okay. So what I thought I would do just to start the course is just give you a sense of what I think are some of the most exciting things that synthetic biology has done. This is sort of according to me. I just think that they're quite impressive. They're both related to medicine, actually. One of them is the biomanufacturing of insulin, and the other is metabolic engineering to produce artemisinin. And so I'll just go through these two stories. Okay, so first we'll start with insulin. So I don't know if you guys are familiar with or know someone who has type 1 diabetes, but the problem you have there is that your body does not make enough of this protein called insulin. And basically this is a problem because insulin, which you can see here is like this little yellow shape, it can bind to an insulin receptor, which is this blue shape, on this orange circle, which is supposed to be an image of a cell. And something that your cells need to do is absorb glucose, basically sugar that they use for energy, from the bloodstream. Right? So you eat food, your body extracts the glucose, and then needs to deliver it to your other organs. So insulin is an important molecule that basically tells your cells, okay, now it is time to absorb this glucose and use it to do whatever the cell needs to do. If you have diabetes, you don't have insulin. And that becomes a problem because then you can't open up this channel and let the glucose inside your cells. So not only are your cells sort of starved for energy, you also have excess stuff in your bloodstream that actually can change the osmolarity of your blood. You'll actually become more and more dehydrated. So this is a problem that makes people quite sick. And I think back in like before there were like treatments, people often could die of this disease if it was serious enough. So there's an exciting alternative though, which is that animals also make insulin. So if you're a human and your body doesn't make insulin, you could get it from an animal. So what I'm showing here, these two ribbony structures that are in the bottom, these are actually the protein structures that scientists have solved for human insulin on the left and cow insulin on the right. And I think what you can see is that they look very, very similar in terms of they're sort of the circles. They have the same number of ribbons and orientations. So they can do the same job that human insulin does. And, you know, we raise a lot of cows already for, I guess people like to eat hamburgers. So it was easier to get insulin from a cow, and then you could inject it and your body would function a little more normally. But there was this problem that it still is a protein from a different animal. It has small differences from the human version. So diabetics who took the basically cow insulin started to develop allergies to them. So they would sort of have a negative side effect of taking this medicine. So the question was, is there a better way to make this protein? So we know that if you're healthy, you have cells in your organ called the pancreas that make this insulin whenever it's needed. But what that means then is that there's a gene, which is basically the DNA that contains the information for how to make this protein that exists in that cell. And so I think kind of what is like the fundamental insight and like the very, very cool idea is that you can put this same gene, these same instructions inside an entirely different cell. In this case, they put it in what's called an E. coli cell. The E is short for Escherichia. People never really know the full name, but basically this is a bacteria that is commonly found in the human gut. There's multiple kinds. There are kinds that make you sick. there are kinds that can live in your gut and you'll be perfectly fine. They're actually probiotics. So in case you might have heard just one of those two, but they were able to take a kind that was not pathogenic, would not make you sick, and basically engineer it to make insulin for you. And what's amazing is because these DNA instructions, even though they came from a human cell, can still be active and produce protein even in an E. coli cell. So you're still able to get this human protein from these cells. There's some missing steps. Basically, what would happen is you would have like a big vat where you'd have your E. coli that are making your insulin protein. So you'd grow it for a while, so they make a lot of them. And then at the end of the process, you need to purify that protein and then send it off, and then it can be injected. But basically, your cells can't make the insulin. We can, with this sort of like bucket of bacteria, make this protein. And what's kind of amazing about this is that this insulin has basically entirely replaced animal insulin. So this is a plot starting back in 1982, showing what percentage of the market share, so of the insulin that was sold, how much of it came from animals versus how much of it was human insulin made by genetically engineered microbes. And you see that there's this shift where it like very quickly took over because people preferred it because they didn't have negative side effects because it was actually the human version of the protein. And something that I think is also really fascinating is that now that we're working in the realm of synthetic biology, right, we can start to think about things that have never existed before but are still good ideas. So for example, if you're a diabetic and you're injecting insulin, maybe you would actually like a different form of insulin, a slightly better one that would stick around longer in your blood, so it would be more effective for longer. And then you wouldn't have to inject yourself quite so many times. And so that's actually what scientists have developed. They now have this analog, sort of like super insulin, that they produce from microbes in the same way, but it can be more effective. And that's apparently now the majority of the market share, which I think is pretty exciting. Okay, so I'll transition to my second story, unless does anybody have any questions so far, or you can put them in the chat or you can unmute. Okay, there'll be plenty of time for discussion later, so no worries. I just want to make sure that I'm not going too fast or being too confusing. It might also be too slow. That would also be okay. All right, okay, so this second, I think, very exciting and cool story from synthetic biology is artemisinin. I remember at the launch event, a lot of people mentioned malaria kind of across the continent being a concern for different people living in Africa. And so this molecule is part of what I think is now the recommended combination therapy for malaria. And it was discovered in 1972 by the scientist who actually won the Nobel Prize for his discovery later in 2015. So this is sort of a small molecule. I don't know if you're familiar with looking at diagrams like this, but basically these sticks are connecting carbon atoms. So it has some hydrogens, it has some oxygens, but basically it's a useful treatment for malaria. And it was discovered actually in a plant. And that is the primary way to sort of get a hold of this molecule. Rather than synthesizing it in a laboratory, you can purify it from a plant, and its name is actually Artemisia annua. The problem with getting a drug from a plant, though, is that how much the drug costs sort of depends on how much of the plant is around. So for instance, if like a lot of Artemisia was planted that year, that means the supply is very plentiful. So it's pretty cheap and easy to get the drug. But say there was some problem, I don't know, there wasn't enough water that year, or the plants didn't grow well, or the farmers just decided they didn't want to plant very much. Then there could be very little plant and therefore very little drug. And so the prices would be very, very high, which is a bit of a problem because this is an incredibly important drug that everybody should have access to if they need it. So the question was, is there a better way to make this drug? And so we can look a little closely at what's happening in the plant. So this drug, artemisinin, is produced by a series of enzymes in the plant that perform very specific chemical reactions. So you can think about, basically, we have here like a series of molecules, and you can see that as we go from one molecule to another, following these arrows, they go through small modifications. Maybe they're losing some kind of chemical group, or like hydroxy is being added, or they're removing some hydrogens. But basically, this is a series of chemical reactions, a bit like a chemical synthesis that's done in a lab, except rather the thing that's doing the synthesis are these enzymes. And each of these arrows represents an enzyme. So that's a protein expressed in the plant that's responsible for these transformations. And it actually makes something called artemisinic acid that then can be converted chemically to artemisinin. Now, the important thing is that the plant is doing this as part of its metabolism. That is like the series of chemical reactions that it's doing to basically collect energy from the energy it collects as light. So the plant gets light, makes sugar, and then converts those sugars into other things. And specifically, this whole process starts with a molecule, FPP, which is farnesyl pyrophosphate. What it means exactly isn't particularly important. The sort of exciting thing is that we can look at another microbe, Saccharomyces cerevisiae, and look at its metabolism and see something very similar. So because a lot of forms of life, whether it's us, whether it's yeast, whether it's E. coli, which I talked about before, all of us express enzymes to break down glucose and recover energy from it, and then also do all kinds of other transformations on these molecules. So it turns out that yeast, when they're processing simple sugars, can also use enzymes to convert molecules one by one, or sort of kind of convert them one step at a time into FPP. Now, you may be like, I've never heard of Saccharomyces cerevisiae. How did they come across it? But this is something that I guess has been sort of a friend to humans for hundreds, if not thousands of years, because this is the microbe that's responsible for helping us make bread. Another thing that yeast do, in addition to making ethanol, is they'll produce gas bubbles while they're kind of fermenting and growing, and that's what makes the bread rise. So basically, this is something that was easy to recover. And then scientists, once they studied the metabolism, so the enzymes that this yeast makes and these conversions that it's able to do, they discovered that it also made FPP. Okay, so why does this matter? What it sort of suggests is that if we could somehow link these two things together, if we could take the yeast half and then add to it the Artemisia plant half of the synthesis, we could go all the way from simple sugars to artemisinin, which would be amazing because then we would have a way of making a drug that didn't require us to grow a plant. And so that is actually what the scientists in this paper that I'm citing in the corner did. This happened in 2006. And it's based on kind of a fundamental idea that's very similar to the insulin story, which is, okay, I know the genes, that is here, these proteins that are responsible for doing this. So I have these enzymes that are proteins. And so if I go into the plant, I can find the DNA sequence or the instructions for making that enzyme. I can put them in a different organism. And because all organisms know how to use DNA to make proteins, they will then produce those enzymes. And then suddenly you've given them the ability to do entirely new chemistry, which includes making the artemisinin. There are a couple more particular steps to this story. For instance, you need to do something to the yeast to make sure that they're doing this pathway, like producing the FPP at a rate that's sufficiently high enough that you actually get a lot of artemisinic acid, but approximately that is the way that it works. Okay, so here's kind of a better summary, I think, of basically how this really cool story for artemisinin plays out is inside the yeast cell, you know that they can take sugars and turn them into FPP using the yeast enzymes. But then if we add some plant enzymes inside this cell, suddenly it can go all the way from sugars into artemisinic acid, which gives us a way to make the drug. And I guess the hope is that at some point, this technology will give us a way to provide the drug without the, I guess, price being very volatile. Sorry, this figure is a little bit grainy, but this is from an article in Nature where they were actually looking back in 2014, sort of at the price over time. So you can see that sort of in the early 2000s, there was a period where this drug was very expensive, but then suddenly it became very cheap, and then it became very expensive again. And it turns out there's actually some complicated economics, which sort of explains why or why not the price has been more or less stable. But people in the field are still very excited about this approach being a way to produce the drug without the price sort of going all over the place. Okay, so I just wanted to summarize. I think there's a lot of technical details in there. And of course, you're in this class to learn some of those technical details. So it's okay if you didn't follow everything. But I just wanted to communicate something about the kinds of problems that I think synthetic biology is really suited for. And what does the solution sort of generally look like? So I think one thing that this really highlights is that the ability to move genes, that are these like DNA instructions, between cells really gives you the power to make these new products. So in the case of insulin, the product was a protein that was expressed by the cells. But you can also do chemistry and get what's called a small molecule, which is there's a small number of atoms. And these are things that often are very valuable as drugs, but can also be something like a biofuel, which would be a gasoline replacement, which would be pretty exciting. Something else I think it highlights is sort of two things you'll hear a lot, I think, if you look at what's being done in synthetic biology these days, or if you're studying sort of historical examples of what's been done, is that there's two sort of, people will call them microbes. What they sort of mean is a single-celled organism. One of them is a bacteria, one of them is a yeast. We'll talk later on in the course about sort of what the important differences are between them, but the main thing to remember is that there are two types. But these are great tools for manufacturing. They sort of came from the environment. One was a microbe that happened to live inside our gut and our like large and small intestine. Another one was a yeast that we had a lot of experience with because cooks have been using it in the kitchen to make bread for quite a long time. But the reasons that they're sort of so useful, I think, is they grow very fast. They can double bacteria can double every 20 minutes. Yeast can double every 2 hours. So they're very effective at producing things quickly. And also the genetic engineering is not too hard. We didn't talk about the details, but the process by which you sort of move DNA from one organism to another is not necessarily straightforward, but it is easier in bacteria and yeast. So that was one reason they were successful. And the last thing is that I think some of the really great strategies that we've seen here, basically so that like biology is very good at doing chemistry. It can take sugars, sort of use enzymes to convert them, and then produce complex molecules. This can be a small molecule or something even like really big, like a protein that has hundreds, if not thousands of atoms in it. Okay, so something I thought would be fun, and I just want to check how many people are on the call. Let's see, and also what time it is. Okay, so it's already 1:40. Maybe I'll give everybody a chance to sort of think quietly by yourself and to try to answer a question. So first of all, I wanted to point out that like from the perspective of science, these stories are very old. The artemisinin one is almost 20 years old at this point, and there's a lot more, I guess, advanced and sophisticated technologies that are available. And so I'm sort of curious about, I think everybody who signed up for this course, maybe there's something that you're particularly excited about or think that synthetic biology would be good at. So maybe take a, I'll set a timer maybe for 2 minutes and just think, what would be a big advance in synthetic biology that you think will happen in the next 100 years and that everyone would be excited about? And I'll do the same exercise. And I guess if we could just get maybe like 5 or 6 examples in the chat, that'd be great. So yeah, everybody take some time to think about this question. In 100 years, what will be the big advance in synthetic biology that everybody is excited about? And then maybe we can discuss together. Oh, awesome. Okay, Maureen has already put something excellent in the chat. She put, or whoever this person is, right? Being able to grow human organs in a lab. I think is amazing and like actually very doable. There are a lot of people who are waiting for liver transplants and heart transplants that it's really difficult. I think that's a great example because it also matches some of what we discussed before, which is that there are certain biological products, like proteins or entire organs, that it's like hard to get access to. But if we could make them in a bioreactor, sort of grow them outside the human body, suddenly they become much more plentiful. Thank you. Thank you, Professor Miriam. Okay, so they put biofuel manufacturing. Again, this is something I think that is incredibly important and also super exciting. You know, we're running out of fossil fuels. They literally come from fossils. They take thousands of years to produce. So the ability to replace some of the molecules that we have to get, you know, by digging deep underground with something we could produce from cells would be amazing. Oh, this is also fantastic. Yes, thank you, Rachel. Being able to create permanent medicines or vaccines for incurable diseases like HIV. Yes. I think I was thinking as well about like the thing that I think about is like gene therapy. Like many diseases sort of arise from the fact that there's sort of a little, I guess, bug in the code or like a small mutation in the DNA that creates some sort of problem. So if you could sort of remove that from the DNA, that would be great. Yeah, awesome. I'm going to take a quick second, sorry, to answer this question from Lillian, who's asking, is there a major difference between what synthetic biology does and what a GMO does? And I think what I would say is one is sort of a subset of the other. Genetically modified organisms are sort of one thing that we can do in synthetic biology. As I sort of said in the beginning, I think if you're engineering DNA, if you're engineering a virus, I think you're still doing synthetic biology. But if you're engineering cells, that would be GMO. And usually when people are talking about GMOs, they're talking about genetically modified crops, which is interesting because plants actually are very hard to engineer, but there have been a couple successes made by companies like Monsanto that are now kind of widely available. So yeah, I think GMOs is definitely sort of a topic within synthetic biology that comes up a lot. Oh, wow, amazing. So many answers in the chat. Okay, so someone else mentioned gene therapy. Being able to reverse aging or prevent aging. I think that would be awesome as well. I feel like that one is potentially hard because we don't understand the biology of aging yet. Like, for instance, what does it mean for a cell to get old? Is it the fact that your DNA has acquired mutations? Is it the fact that you have sort of collected a bunch of misfolded and old proteins that sort of block the behavior of the other ones? But definitely there's a bunch of people working on it. I don't know if you guys, I mean, I'm sure you know about the company Google, but they actually, they fund an entire company called Calico Labs that is entirely looking at aging to understand it so they can reverse it. Someone wrote enhanced tumor destroying cells, 100%. I'm sure you guys, or maybe you've heard of these things called CAR T cells, because they're kind of a modified T cell, which is something from the immune system that can basically go to the tumors and attack them. Sustainable agriculture and food production. Yes, I mean, I love that answer as well, because it's super interesting to think about, again, biology that hasn't existed before. You know, like the climate is changing, and I like it's going to affect a lot of places, particularly parts of Africa more than other places. And we still need to be able to grow plants. But there's like, you know, evolution is powerful. We can create new types of plants that maybe will still be productive in that space. In addition to the problem of like, we just want to produce more food per square meter than we have before, which would also require an entirely new plant. Okay, okay, degenerative diseases, Parkinson's and Alzheimer's, great. Oh, Kato finally joined us, amazing, thank you. I'm sorry, it was so hard. Yeah, more resistant plant cultivars, improved crop yield, yes, yes, yes. Genetically engineering fungi and microbe to increase their efficiency as biocontrols, which will reduce use of chemicals. Ooh, yes, that's also quite nice as well. Because, I mean, we use a lot of pesticides to make agriculture reliable, but could you somehow engineer a microbe or a fungus to maybe replace some of those pesticides? I don't know if that's what this person is referring to, but that's definitely one example of it. Oh, Masuda said we may be able to use harmful organisms or microorganisms to produce potential products. Yeah, I think this is interesting. I mean, I'm not sure quite what you mean by harmful organisms, but definitely people are excited about what's called a non-model organism. These days, people are really excited about working with types of bacteria and yeast that people haven't worked with before, because they do, for example, entirely different chemistry. So there's people who are trying to study, or I guess rather discover new medicines by just looking at what are all the enzymes that exist in all the fungi that are sort of like in the soil of a forest somewhere, because all kinds of cool things. Animal production, milk, meat, disease resistant, yes. Biological computing as in bioinformatics systems. I think this is super exciting. Yeah, we haven't really talked too much about like the math and computer science sides of this, but I think one, being able to use cells as computers is pretty exciting, but also kind of, can we use mathematical tools to help us like kind of learn how biology works, so we can create better designs. Genetics for disease diagnosis, yes. Reversing genetically, yes, yes, yes. Synthetic food like, yes, yes. So I guess, yeah, this Martin mentioned synthetic food like meat. So I guess, yeah, it takes a lot of water to just raise a couple cows. So if we all want to keep eating hamburgers, is there some way to produce meat that doesn't require, growing and feeding a whole cow? Yeah, making more drugs. Yeah, with recombinant DNA technology. Yeah, that's basically it. I guess Martin has used an interesting phrase here, recombinant DNA technology. That's actually a good name for what we basically talked about. Recombinant DNA basically means you've recombined the DNA. So that's this process of like taking a gene from one place and putting it in a new place. And then Moses put vaccine production. Yeah, I also like that as well. I think it's interesting because it's quite easy to get really excited about the science, but also there's a lot of engineering that we need to do. Also in terms of like, how do I build a factory that can sort of scale up some of these processes, so I can go from making maybe like milligrams of drug sort of in a laboratory setting, you have this like tiny tube, versus I need to make kilograms of drug in like a bioreactor that has like hundreds to thousands of liters in it. Yeah, awesome. Thank you guys so much. Oh, sorry, I think, is someone's microphone off? Do you want to say something? Oh, okay, awesome. Yeah, plant species. Okay, wow, this is amazing. I feel like we could just talk about this for the rest of the class, but I can see you guys are really creative and pretty well versed, I think, in like some of these ideas already. So I hope that the course can like give you the tools. So if you like want to learn more about these things, you can. Okay, Kato, I saw your message about taking a photo. Would it be okay if we waited until the lecture was done to take the picture? Okay, awesome. Please bring it up when the lecture's over and we'll do it. Okay. Thank you guys so much for engaging. That was a super fun discussion and I look forward to chatting more. Okay. Oh, Ibrahim, that's a great question. I think we'll definitely get to the challenges in a later stage. But I guess I could briefly, I guess, summarize what I think some of the challenges have been, at least from the previous two examples. So I think one challenge is, so I gave this example of like making artemisinin. Sometimes you can put in all the enzymes that because of what you've like written down on the piece of paper and you've studied the chemistry, should be able to do all of these steps, but the cell just doesn't do it. And it turns out that the answer is sometimes that it's actually sort of, the cell is trying to balance. It can only do so many chemical transformations at one time because it only has so many enzymes, so many molecules. So it's actually putting a lot of its energy towards like growing and dividing, because that's normally what cells do, rather than making the thing that you want. And so it can be incredibly difficult to sort of like not make your cell sick while still giving it the ability to sort of have this entirely new behavior. I think another challenge is just that if you want to work in a more complicated organism, so something that's not a bacteria and not a yeast, so say you want to engineer a human cell, say you want to engineer a plant cell, it's actually really hard because we have a lot of systems for sort of recognizing, like for instance, humans need to protect themselves from viruses and also bacteria, right? Because these give us infections. And we can detect that they're present because there's excess DNA that we don't recognize. So I think there's a lot of systems to like chew up DNA or just keep it from being integrated into the genome. So it turns out that some cells are actually much harder to genetically engineer than others. And I think also on the medicine side, we really don't understand immunology very well yet. And so we've created some like really exciting medicines, but then you put them in the human body and they just don't do what they should do. So I think that's a great question, and I'd love to give a longer, thoughtful answer. But yeah, I think those are some of the things that come to mind. Okay, I actually wanted to have a brief poll as well. Let me see if I am good enough with Zoom that I am able to do it. Let's see. Okay, great. So I think, do you guys see something called a biology background poll? There's just 2 questions. And basically, I wanted to get a sense of sort of like how much biology have people done before? What's your like level of comfort and something you're excited about? This is just to help us because we're sort of developing the other 5 weeks of the course. So we can figure out like what's information that you would like to learn and also what you're excited about, so we can sort of focus on your interests. While you guys are taking time to respond to the poll, I just wanted to give also a summary of, again, some of the statistics from the sign up form that I thought were interesting. So we have quite a mix of people's educational backgrounds. We were sort of imagining this would mostly target college students, and it seems like at least almost 2/3 of you are undergraduate students. But then maybe about 1/5 are master's students, and then maybe 1/10 are PhD students. So that's really exciting. I hope we can have something that will be suitable for all of you. I was also curious because, we're targeting sort of any student in Africa, but it's quite a large place. So sort of where are people coming from? So it looks like, again, we have the most people signing up from Uganda, but then also a lot from Kenya, Nigeria, Ghana, and Tanzania. So I thought this was really exciting. The only place I've been to on this list is Ghana. I got to visit for a couple of weeks in college, and we went to Accra. But it's really cool to meet people from all these different places, and I hope you enjoy interacting with each other. And the last thing I was interested in is like, what are people studying? And kind of what's your background and interest? And I think we see like a lot of different topics. So the big ones are a lot of people, it seems, are studying laboratory technology, a lot of people studying molecular biology, which I think is like a really nice fit for synthetic biology, a lot of biotechnology, people who have something that's like called biomedical in their background, also biochemistry. But then there's also some, I think at least one person who signed up has a business background, maybe someone with economics background, people studying genetics and medicine, plants and zoology. It's really exciting kind of how broad the background is. Okay, so it looks like, wow, almost 70% have filled this out. So that's great. I'm just gonna take a quick look at what people have said so far. So it seems like, okay, most people, which I guess this is consistent with the sign up form, are have taken a biology class and are like studying something related to biology, which is great. And then a few people doing engineering and something else. Okay, some of you have done iGEM, and then some of you have worked in a lab, which maybe makes sense if you're studying laboratory technology. And then in terms of interest, this is quite spread. This is nice. So, okay, so I guess almost half interested in human health, which of course, very interesting, but also improving the environment, crops and agriculture, and then some people excited about the bioeconomy. Awesome, this looks great. I think this is kind of aligned. I mean, I got these ideas also because, you know, I work in a synthetic biology lab for my job, and have a lot of friends who are very excited, or coworkers. And these are also the kinds of things that we enjoy talking about. So it's super cool. All right, so let me just check the time really quick. Okay, so it's almost, well, I guess it's different times for everybody. It's almost 2 p.m. for me. Oh, awesome, people are putting, yeah. Okay, so a lot of biotechnology and molecular biology, like biology, laboratory technology, and crop science. Okay, looks great. Okay, I'll leave the poll open just in case, but I was gonna go ahead with some of the next slides. I can close that. I'll leave the chat open. So one of the reasons I wanted to ask about your background was then just to tell you that sort of, I think no matter what you're interested in, there's sort of a cool intersection with synthetic biology. So I just went to this journal called ACS Synthetic Biology, which publishes synthetic biology papers, and just looked for things that have been published in the last year to just give you a sense of kind of how broad it is. So for instance, here's a recent paper that's looking at how to automate the design of genetic circuits. And this is like only a math paper. They did no experiments. They just wrote down equations for how genetic circuits worked. They used something called a global mixed integer optimization algorithm, which sounds very fancy, but we're able to produce this tool. But then also, if you're interested in chemistry or material science, there's cool work to be done. So these people there was basically, this is another, I guess, small molecule, but if you shine a particular wavelength of light on it, will actually change shape. So if you incorporate this molecule inside a protein and then grow it into like a larger hydrogel, it's basically like a squishy substance, sort of how it responds to temperature will vary based on whether or not you've shined a light on it. So basically you have now this like light responsive material that you've encoded using biological materials. If you have a background in electrical engineering, actually that's also really great. I think synthetic biology gets a lot of inspiration from electrical engineering. And you'll notice that we call gene circuits circuits, and it's because it makes people think about electrical circuits. The idea that you have components in electrical engineering, it's like capacitors or transistors or resistors, and you sort of wire them together and then they do complicated logic. A lot of people think about a cell in the same way, which you can see here. Okay, we got many more examples, but I think as kind of came up in the discussion earlier, if you're interested in immunology and human health, there are people who are engineering immune cells to sort of change how they respond to disease. If you're interested in biomedical engineering, which has some overlap, but basically the idea is maybe you want to like build a tissue outside the body. A lot of people are working on problems related to that. Also, if you're interested in maybe like public policy or business, actually, I think last year, President Joe Biden signed in like a big bill to sort of fund what was called the bioeconomy, because we need people to think about how do you give money to researchers or to companies in order to encourage them to produce the best ideas. And this is sort of like a business and politics question. Okay, this is the last slide, but yeah, if you're excited about crops and agriculture, again, people in synthetic biology are looking at engineering plants, which is pretty exciting, even though it is challenging. So my background is actually chemical engineering. So this one sort of spoke to me, but there are people who are looking at how do we maybe engineer yeast so that we can take leftover biomass from other processes and then turn it into valuable chemicals like farnesene. This is sort of a cartoon of that process. And then as the last one, say for instance, if you're interested in physics, there's a lot of cool physics, both designing sort of like microscopes so they can read or observe interesting properties about cells, but maybe also studying things like an ion-gated channel, where whether or not this sort of protein complex is open or closed depends on, I guess, electrical current or application of some kind of axon potential in a neuron. It's all very exciting. So that's kind of a lot of examples. You could look at the slides later if you think any of these are particularly interesting. Again, these are like brand new research articles, so they're quite technical, but I think they just show that, you know, even in this journal that has synthetic biology in its name, there's a lot of different things that people are talking about. Okay, so now we'll do some just quick kind of logistics for the course, and basically I think some advice on how to get the most out of it. So if you can join the lecture on Wednesday, which you all are doing, that's great. If you can't, if you can watch it later, that'd be great. We're going to send out a link over email. It'll either take you to the Google Drive or an unlisted YouTube link, but please watch the lecture as a way to get introduced to the material. Also complete the homework. The homework is sort of the way you like practice, and I guess as you know, or just sort of like check that you've actually understood what you've learned about in the class. As I'm going to send a link in the chat, and it'll also be later on, but we already have the homework for week 1. And the idea is to use the second half of the class period. I guess we started a little late today, but that's okay. But we'll work together on the homework, sort of at least for like 20 to 30 minutes together. And then you have the rest of the week to finish it. And then also something I think is really helpful is like talking about the homework with other people. Another way to check how much you understand is to see if you can help each other or explain your answers to each other. So again, we'll hang around until like the second half of the class period to work on things. But also we have, like I can put you in breakout rooms in Zoom, or you can get on Discord, and Discord gives you, there's both like a video chat and an audio chat and like a just typing chat option. Okay. And just to give you kind of an overview of what's gonna come. So week 1 was just sort of like introduction to the topic. This is why it's exciting. What is synthetic biology? But then going forward, I think, so Steve Millennia, who is on the call, I believe, will be teaching next week. So he's going to talk about how do cells make things. Basically the, oh, someone just asked in the chat about resharing the link. We will definitely do that. Okay, sorry. So since a lot of synthetic biology is about engineering cells, that's kind of what we're going to focus on. So Steve will talk about how cells make things. The next week, once we sort of understand that biology, we will then ask about how to control what cells make. If we kind of understand the way we do it, we can then perturb it and sort of get it to do something different. Then on week 4, we'll talk about sort of common components or parts in synthetic biology. So these are like proteins, pieces of DNA, just the kind of things that would show up if you were gonna want to like build your own circuit or engineer a cell. And then on week 5, we'll try to get a little more practical and talk about what does synthetic biology look like in the lab? How would you actually do some of these techniques? And then the 6th week, it'll be kind of special. We're going to do something called cell biology by the numbers, which is sort of like an interesting way to think about, say, how big is a DNA molecule or a protein molecule? How big is a bacterium relative to a yeast cell, for example? But we're also going to discuss any topics that you guys are excited about. So there's actually a question on the homework, and you get credit for it if you answer it, about what you would like us to talk about in week 6. So you can think about what you want to learn more about. Okay, so as the final part of the lecture, I'm going to teach you hopefully a little bit of biology. And it sounds like many of you are studying biology, so apologies if this is old for you, but hopefully if there are some people on the call who this is totally new, we're going to go through it. And I guess I just wanted to say that I think biology is sort of different from a field like physics, which is really focusing on like unifying principles and saying, you know, there's just a small number of things to keep track of that are important. In biology, I feel like it's all about diversity and complexity. Like there are so many types of life on Earth. There's so many types of proteins that have evolved. So I think that can make it kind of daunting because you're like, to learn biology, I must memorize a large list of things. And it does help if you know things on the list, but there are certain tricks you can use to sort of simplify this process so it doesn't feel quite so overwhelming. So my first tip to you is to tell you that every molecule we learn about is going to be, for the most part, just one of 4 types. There's 4 major types of molecules that show up in biological systems. So anytime you learn a new word, you can just sort of ask yourself, okay, this thing that we're talking about, it's got to be one of these 4 types. So the first type is a nucleic acid. This is the Na in DNA and RNA. The second is proteins. So this is sort of the molecules or the machines that are made by the cell and sort of do a lot of its work. Examples are things like enzymes, or you'll learn about later, transcription factors. The third one is carbohydrates, or another way of saying this is sugars. Maybe you've heard of glucose or cellulose. These are both examples of carbohydrates. And then the last type is lipids. And so you may have heard of phospholipids or triglycerides. And basically these 4 types of molecules, they have different chemistries. We'll talk about that later. You can understand how they work even if you don't know their chemistry. But they play sort of different roles for the cell. So nucleic acids tend to store information. Basically, there's sort of a sequence of these nucleotides, which we'll talk about, which hold the like instructions for how to make proteins for the most part. Everything you need to know, for example, about how to produce a cell that's in your body is encoded in your genome, which is the long set of DNA that's inside every cell. The proteins I think of as sort of the machines. These are the things that are doing the chemistry, binding to other things, and I guess turning on gene expression and doing gene regulation. Carbohydrates and sugars tend to mostly be for energy, which makes sense. We need to eat to get the glucose, to have energy to do what we do. So they largely do that, although some of them are structural. So cellulose is a long chain of sugar molecules that's in the cell wall of plants and makes them rigid. Lipids, it's similar, but a little bit different. So there's another like colloquial way to refer to this is a fat. It has a particular type of chemistry, but this makes it good for structure. So the walls of the cell are all made of phospholipids. All cells have a cell membrane that usually contains phospholipids, but they can also be energy. We also get energy from the fats in our foods, like triglycerides. And to make things even simpler, I think 90% of the molecules we'll learn about in this course will either be nucleic acids or proteins. So these are really the things to pay attention to. And I'll just go into a little more detail about what those things are, how they work. So first, nucleotides. So nucleotides are polymers, which is another way of saying they're long chains of repeated units. Usually when people say polymers, they think of plastics because plastics also are long chains of repeated units. But here, this is a polymer of something called nucleotides. Nucleotides have 3 components. Again, this is just sort of defining the chemistry. A phosphate, which includes phosphorus, a sugar, and a base. And here's actually a picture of what they look like. So you can see that there's 3 parts. There's this charged phosphate group, this sugar called deoxyribose, if it's DNA. That's the D in DNA is for deoxyribose nucleic acid. And then the base. And there's actually 4 options for the base. They have particular names, but people usually refer to them as A, T, G, and C. If you've ever looked at a sequencing file, you've probably seen this, and that's actually what these letters stand for, okay? So if we chain these things together, now we have an example of a DNA molecule. So this is a polymer of nucleotides. And you see, why does having that phosphate even matter? It's because it gives these things the ability to stick together and then form this long backbone that connects them in a chain. So for example, this DNA chain is 4 nucleotides long. But something that's particularly cool about DNA is that these particular nucleotides, again, because of their chemical shapes, can form hydrogen bonds. So I don't know if you guys remember from your chemistry courses, but this is a kind of bond. It is sort of less permanent than, say, a covalent ionic bond, but it's similarly mediated by electrostatics, or the fact that negatively charged things are attracted to positively charged things. And we see that here. So usually the hydrogen, I believe, is positively charged, and then the oxygen is negatively charged, and they bind. But the main thing is that there are these these dotted gray sticks. And so now I've labeled them, and you can see that actually the 2 base pairs that bind with each other are very specific, because you see these shapes sort of fit together almost like puzzle pieces. So A sticks to T, and what's special about their interaction is they just have 2 of these hydrogen bonds. We see a hydrogen here and a hydrogen here. And we see the same thing here. It's T binding to A, they match up. And then G always binds with C. And here there are 3 hydrogen bonds, 1, 2, 3. Something that's kind of interesting is that actually the connection between G and C is a little bit stronger and sort of like less floppy than the AT binding. Okay, so I think what's cool about base pairing is that now it gives the ability for, not only is it sort of like there's information in there now, if you sort of read along the DNA sequence, you'll get a set of letters, that's the way that it encodes the information. Is you might have like A, T, G, C, C, T, G, A, T, G, like some set of information like that. But base pairing also means that you can get interesting shapes when in 3 dimensions when the molecules stick together. So DNA is usually what we call double-stranded. So there's these 2 strands stuck together. And it's normally in a shape called a double helix, which basically means you have these 2 strands. You could think of it almost if you had like a ladder that was flexible and then you twisted it, you would get a shape like this. And here, I think this previous slide is like very specific and has like all of the atoms drawn. But usually in synthetic biology, we don't need to know all the chemistry. You just need to know that A binds to T. So you might see pictures that look more like this, right? Like they've not drawn the phosphorus groups. They've not drawn the hydrogen bonds, but what they want you to know is that A can bind to T and G can bind to C. Okay, so there's this double helix shape, which is really nice. The other thing I think is cool is that there are also RNA molecules. So the R stands for ribonucleic acid. It's just a different sugar in the nucleotide backbone. But it can, as an individual strand, sort of fold in on itself. And then the bases can also pair with each other. And then you get interesting shapes like this. This is called a stem loop because it has a stem and a loop. And one reason I think this structure is exciting is that if you know about CRISPR-Cas9, for example, which is a tool for genome editing, an important part of sort of that system is something called the guide RNA. And it also has a stem loop structure, which is important so the protein can bind onto the stem and they can travel together. But it's pretty much the same concept. You see here, so RNA is slightly different because U's are T's, but you see we have C's and G's with a line between them showing that they're base pairing. And now you can predict their structure. Okay, well, we're running out of time, but all right, so I'll just quickly talk about proteins. So proteins are very similar in that they're also long chains of repeated units, except the repeated unit is something called an amino acid. It has an amino group, a side chain, and a carboxyl group. And approximately the chemistry looks like this. This is a slightly different cartoon style, but you see we have similar atoms. We have nitrogen atoms, carbon atoms, hydrogens, and oxygens. And here, the part that can change, and before it was the base, we could choose A, T, G, or C. Here it's something called the side chain. And actually there are many, many more options for proteins. So there were only 4 types of nucleotides, but now there's 21 amino acids. And again, it can be important if you're taking a biochemistry class to memorize these, maybe to be able to draw them. But I think the main takeaway is that these side chains have different sizes and they have different chemistry. That is, some of them you see have this plus sign indicating that they are positively charged. Some of them have a negative sign indicating that they're negatively charged. Some of them are uncharged and polar. And this chemistry just sort of affects how this long strand of repeated units also will fold and the chemistry it has, particularly if it's going to catalyze some kind of chemical reaction. In the same way, the fact that this kind of basic unit, the amino acid, has these 2 groups gives it the ability to produce a bond so that you can like put them together in a long chain. Here it's an amino group reacting with a carboxyl group, but again, the chemistry is not particularly important. But I wanted to highlight here, this is an example from actually a nice set of blog posts by this group Khan Academy. I can send the link later. I'm basically showing there's a large set of things you can make with proteins. So there are digestive enzymes, which can break down nutrients. They can do transport like hemoglobin, which carries oxygen in the blood. Some of them are just for structure. Some of them are for hormone signaling. So remember we learned about insulin. That's kind of a signaling protein, a protein that carries information. Antibodies, which are for defense, and sort of many other properties. So proteins, I think, again, they perform many diverse and essential cell functions. So yeah, it's quite exciting. Okay, yeah, so again, this will be kind of important for lecture 2, where we're gonna get into more of the details of sort of what biology these things can do. But yeah, just remember, you can always ask yourself, okay, this new thing that I learned about, is it a nucleic acid? Is it a protein? Is it a carbohydrate and a lipid? And hopefully that will help. Okay, so we've reached the end of my notes. I'm sorry, it took a bit longer than I was expecting. But yeah, if anybody has any really big questions, you can unmute to ask. You could type it in the chat, but I will be available on Discord to answer questions later if you want to post them there. So maybe I'll take 1 or 2 questions if someone has something that they really want to ask. Okay, well, it's fine if it takes time to kind of formulate your question. Yeah, maybe, I guess many of you are in biology, so probably this is not new to you, so I hope it was clear. All right, so the plan now is we'll transition to a time where we can work on the homework together. You're welcome to stay on the call. If you want to be put in a breakout room with some other students in the course, I can put you in random groups, in like groups of 5. You can ask about the homework, but I guess I'm gonna stop screen sharing. And oops, okay, here is a link to the homework if you want to open it. Okay. Okay, I see there's one question, so I'll go ahead and address that. Okay, please comment on intended effects of knockouts using CRISPR. Okay, so I feel like I have 2 ways of interpreting this question. So one is like maybe you're asking, so say you've used CRISPR to knock out a gene and it hasn't had the effect that you expected. That could be one thing, which I think is quite common because what goes on in a cell is very, very complicated. I think a good analogy for what biologists are doing is trying to understand how a plane works by just being given a plane and then sort of trying to take it apart and see how it works. So sometimes people make a perturbation and then the effect it has is unexpected. I think though you might also be asking about, I think one of the primary concerns around the safety of CRISPR is say that you use it to modify a piece of DNA and then you accidentally modify something else. And yeah, I guess the mechanism by which that happens is just, so with CRISPR, you have an enzyme that can cut DNA and you use the guide RNA to basically take this DNA cutting enzyme and take it to a particular place in the genome. But you might tell it to go here, but then like maybe a very small fraction of the time, like 1% of the time, it actually goes somewhere else. And then that can be a problem if you say are editing something very important, which is like a therapy, like an immune cell that you want to put in someone's body. And it's also why people are very, very scared to edit human embryos. And I think it's actually illegal almost everywhere in the world, because you sort of don't know what you're doing, and it would be heritable, so you shouldn't do it. I hope that answers your question. I think if people are excited about learning more about CRISPR-Cas9, we put it tentatively, I think, in week 4. So that's definitely something we can talk about more. Let's see. I thought as a last thing, we can go over the homework together because we're doing something that maybe is different than what you have done in other courses. Since the course is fully online, we're going to kind of take the homework in over Google Forms. So I don't know if anybody has opened it, but this is what it looks like if you click on the link. So it takes you to a Google form, and there are a set of questions. So I actually already, if you see this here, there's already a link. If you click on this button, if you click on here, it'll automatically take you to the slides that I just presented. I'll send a link, of course, but yeah, feel free to just, you kind of already have access if you want it. The other point I wanted to make is that we sort of designed the assignments in a different way, and that you can basically, there's a set of easy questions. There are some medium difficulty questions and some hard questions. And the idea is like, you basically get to decide, sort of like a video game, what level you want to play the course at. So you can play it at a beginner level, you can play it at a medium level, you played it at an advanced level. All you need to do is earn 5 points total. So you could, each of the easy questions are worth 1 point. So you could do all the easy questions and 1 medium question, and you already have full credit. However, the hard ones are worth 5 points. So you could just invest maybe a little bit extra time, just do 1 hard question, and you would also get full credit. If you want to, though, you can just do all of them, because that would sort of maximize what you're able to learn in the course, and we'll still give you sort of a grade higher than 5. I just wanted to note that the hard questions might take a little bit of extra research. Also, because again, we're doing this online, people are joining from all over the world. We understand that you might not be able to turn the homework in when it's due, and it's totally fine. So ideally, you'll have submitted before the next time we meet. So that would be before next Wednesday. But basically, feel free to submit it whenever it's done. Just notice that if you send it in late, it'll get graded late. And so then whenever we do the certificates at the end of the course, it might come in a little later. But, you know, kind of pick something that suits your schedule. Also, it's very, very important that you kind of use an email address here that's the same email address where you receive notifications about the course, so we can be sure to give you a grade. Because if you turn this in and the email address is wrong, I won't know who to give the really good grade to. So please put your name on it. Yeah, does anybody have any questions about kind of how we're going to manage the homework? Or maybe you have it open and you have a question about something that we've asked you to do. Oh, I see a question. Can we get that link shared on Discord later? Yes. And then how are we getting your groups? So feel free to like self-organize. Like something you could do is just go on Discord later and type in the general chat, like, I am looking for some friends to do homework with. Does anyone want to join me? And you could like make your own Discord channel and hang out there. If we have more time during the lecture, I think I can also put you sort of in random breakout rooms. We could even do it for like 10 minutes now if people liked. Yeah, if that sounds good. Yes, we will send. Yes, and the recording will also be sent out. Did anyone, so I sent a link to the recording for the launch. Did people receive that? Did anybody open it? Did it work? Okay, so some people saying yes, it did. Okay, fantastic. Oh, okay. Okay, I see your question, Masood. Actually, I can look up what email address you're using and email it to you. Let me just make a note to myself, and so I can make sure I've answered everybody's questions. Okay, so things that I should do after this class, I'm going to send out the recording. I'm going to send out a link to the homework. I'm going to send everything on email and on Discord. And then I'm going to write down this person's name, Masood, and then I will tell you what email address you're using. And you can also contact us if you have a personal question. Feel free to message me on Discord, or you can use the SynBio4All Gmail address. I check that, Kato checks that. Yeah, again, we'll just try to answer as soon as we can. Any other questions, or yeah, anything that feels confusing? Okay, well, I know that silence can either mean everybody, oh, what next after this course? Oh, that's a great question. We'll do another one. So yeah, we'll have an intermediate level course. But yeah, we kind of need your help. You guys are sort of our experiment class, and we kind of need to learn how to manage something that's like fully online. I think maybe before everyone leaves then, it would be awesome to do another group photo. We did one after the launch and it was really fun. So, and I even think it was like maybe it was like something in the app. Oh, Brenda asked, I'm not going to teach again. No, but I'll be around. I'm going to help for office hours. We have some other great folks. I'm particularly excited because I think people who've worked on the iGEM teams or are like trained in bioinformatics, and like Steve, I know is like working in Malawi, are going to teach. So yeah, I think it'll be really fun. Okay, so Are people excited about a group photo? I don't remember how to do this. Okay, I think I'll just take a screenshot. So whoever is comfortable, do you want to turn on your camera? Oh, hello, Martha. Wow, it's really nice to see you guys. Amazing. Oh, Alex, the guy with the Friends background. Are you a Matthew Perry fan? Is that why you have the Friends background? Oh, I'm sorry. It's a sad time. Hello. Oh gosh, this is so cool. Okay. Okay. All right. Oh, there's Kato. He looks cold in Paris. It's cold in Boston. Okay, my friends. I'm gonna count down if you're ready. Okay, 3, 2, 1. Let's go. Yay! All right. Yeah, well, it was a real pleasure. Thank you guys for joining. We'll be back at the same time next week. It's the same link. We'll send it out again. Hopefully there aren't as many problems. But yeah, let us know if you have questions. Please do the homework. I'm gonna hang out for at least 10 more minutes if anybody wants to talk about the homework or ask something about the class, but feel free to go. Everything will come out over email and on
Thank
Discord.
you.
Yeah.